Okay, so if you've gotten up to this point and you've evaluated that motion registration worked really well, as I said, but your own two eyes are the best benchmark and detector, because we have these amazing visual systems that really let us see very small movements. And so if you're really happy that the registration has worked really well, now you have every pixel for your entire recording should represent the exact same cell, the same position in that cell, so that we can kind of forget about motion and start to look at correlation between those pixels to try to find which ones of them go up and down together. So that's cell detection. We'll get to spike deconvolution if we have time. We'll make time. We'll talk about it when we get to it, and then the quality control. This is really what we call loading up the data in the GUI and learning to look at it from the different visualizations that we provide. Again, to get a sense, every step of the way, you want to have a sense that you know how well this is working and what are the things it's failing at. And so that's why Carson has built this GUI that, as you'll see, and you've already using it probably, it really lets you get up close to your data, right? So let's see. So we start. So what is what is going to help us here to detect cells is going to be the activity of these cells. So we're not just taking an anatomical stack of some GFP. We're taking usually a GCamp or another time-dependent calcium sensor, and so at different times, different cells are active. We're going to take advantage of that. The thing that we're actually taking most advantage of is just how almost apparently randomly these cells are lighting up at different times. This is actually it's one of the best properties of these movies, is the fact that neurons have this kind of noisy aspect to them, that they fire action potentials. Actually, in this case, this is in visual cortex, and we show the same movies twice, the exact same. We show the exact same sequence of stimuli twice. We got completely different patterns of activity. So we're actually taking advantage of that when we do cell detection, because it means that we can pretty much, if we see a kind of a group of pixels light up at one time, because cells are so sparsely active, it's unlikely that the nearby cells are active too. And so even just seeing it once on a single frame is pretty much enough for kind of us to kind of convince us, okay, there's a cell there. I just saw it lit up, right? So we built the algorithms to kind of take that into account, basically. This has been, we started with a more complicated approach that I'm not going to tell you about, but really kind of the latest version of suite2p that this has evolved into is pretty much trying to mimic what we do when we look at these movies and say, something popped up, there's got to be a cell there. I'm just going to draw a little mask around it. It's doing almost just that.
But the cell is not active at all.
We'll talk about what to do in that case. Yes. Let's get to it. This is the actual movie you'll be using. You saw this from Carson already. Good. Here I just have more examples of data. So this is a mesoscope movie where these are ROIs all over the field of view. Now, there's nothing special or different about this. I'm just recording it in different parts of the mouse cortex. Even different brain areas, as long as they're kind of on cortex, they're all pretty similar. Things can get a little bit different if you're recording subcortically for all sorts of reasons. First, because you have to get there with your microscope, so you might have to put a green lens through and so on and so forth. So things can get a little bit different in terms of the statistics of these movies subcortically, but cortically, and as long as you have an action potential dependent sensor, the problem is pretty much the same problem. It's the data sets do not vary hugely, at least in mouse. And even if you're going to a different species, well, zebrafish, you'll have maybe something with very dense labeling of cells. That will be maybe similar to like mouse hippocampus, where things are also quite dense, the cells are. So that's maybe a slight change, and actually there's a whole team we put together, Team Dense Recordings, that's just to kind of investigate, what are the best settings to use in that situation. And okay, if you're doing file recordings, that's a whole different thing. We're not going to talk about that today. Today, it's mostly going to be about somata, and it's going to be about axons and dendrites. Well, next 3 days. Okay, again, just another way to get a lot of cells. I should also say this is the reason we kind of started with this in the first place with suite2p, is that we were trying to do these kind of very large-scale recordings, and so we wanted to minimize as much as possible the amount of manual curation we'd have to do on these data sets. So the algorithms had to be fairly good, but also had to be fairly fast, because we didn't want to wait forever or like have to use big computing resources to run them. I mean, we can record, a single person can record like 3 big data sets like this in a day, and then if we have to wait another 2 weeks before they get processed, they're kind of, would slow things down. So currently, suite2p can process data set up to this size, maybe in about as long as it took to record them. So it kind of really, it allows us to do this kind of, let's say, closed loop science, right? Where we go on one day, we do a recording like this, we let it process for a few hours in suite2p, and then maybe in the afternoon or evening we start analyzing those results, get a sense of what happened during the recording, maybe use some of the visualizations we built into suite2p that we're going to tell you about later, and then get a sense, oh, how should I change my stimuli or try to change my behavior to get a better experiment. So that's really what we primarily developed this for, but of course along the way, as I said, since everyone's data sets are not that different, it turned out it's a good tool to use for in all sorts of scenarios. Okay, and if it works, we get something nice like this from a zoomed-in recording where you have relatively many pixels per cells. You'll get things that are kind of more pretty like this, where the GCamp is very clearly just kind of in the cytoplasm. There's this big nucleus in the center. I'm always surprised just how big these nuclei are, right? But yeah, most of that, the dark place is the nucleus. We color the cells with random colors just so you can tell them apart when they're next to each other. And that's what they look like from one of the big recordings. A whole lot of ROIs there. Okay, this is what we're trying to get. We're trying to get a set of masks, which essentially all we're going to do with these masks is we're going to multiply them with every single frame of our movies to extract our fluorescence traces. So if you wanted to, you could weigh all these pixels equally, and there would just literally be an average of the pixels inside the masks. But we want to take into account the fact that some pixels are very dim, either because they're in like the nucleus or maybe they're like on a dendrite. So we want to weigh those less because they have less signal. And so there's still positive weights, but it's a weighted average of the pixels inside that ROI. And that's by and large going to be our extraction method once we find these ROIs. It's not going to be anything more fancy than that. So how do we find these masks? Okay, some ideas. What I kind of wanted to roughly distinguish into 2 classes, structural and functional ways of doing cell detection. Now, one thing you could do, and I think this is maybe like the classical way, is you have some kind of structural image that you can go by and just kind of pick your neurons, by hand one by one, maybe circle them. Maybe you'll just make circles. Maybe you'll be very careful to, exactly follow the boundaries. So this, this is something you could like do in like Fiji or ImageJ and very easily. You could do that based on the mean image alone. So just you register the movie and you just take the average. Or you can get more fancy. Nuclear tag would be really fancy if you like really wanted to put a nuclear tag with your GCamp, then you could perhaps actually use one of the automated algorithms in Fiji to detect those nuclear tags, because they're just blobs, and they have algorithms for detecting blobs pretty easily. And then you could take some bigger circle around those blobs. Or you could go in by hand and put in the hours to trace all of them. I guess what I meant by you can get more fancy than the mean image is you can use something like the variance image. So you could, instead of averaging over time, you could take this variance or standard deviation of the movie across time. That will give you a sense of which pixels were the most active because they changed the most across time. So that is kind of already going into kind of more functional methods because you're already using something functional about it, but you're still kind of summarizing it just with this one image that you may either by hand or with an algorithm try to detect otherwise. Or you can take things like skewness across time. For those of you that know what skewness is, so this basically measures something about how often do you get an outlier in your pixel activity across time. And this is good for calcium traces because they tend to be flat most of the time, and they just have a few times during your recording where there's events, and so those are the outliers. And so if you take skewness alone, that might give you an even better sense of which cells were sparsely active during the recording. And so I would categorize all of those under structural because they rely on a single summary image of your recording. And now the class of functional cells is a lot larger. Functional algorithms is a lot larger. It almost only includes automated algorithms, all sorts of pipelines that people have developed over the years. Now, okay, so we already talked about using a single image, but I haven't told you, and we've told you variance, skewness. Okay, maximum projections I haven't mentioned either. So that's actually even more useful, we found, than these other two because of this property that if you see a cell that's active highly, even on a single frame during the recording, there's a cell there. So if you just take the maximum, and that maximum is really high for one frame for one cell, it's going to really pop out of the kind of the background, which doesn't have these extreme time points. And then there's things like what we call a pixel correlation map, which is where, again, just a single image, but you make it by correlating every pixel with the average of the pixels around it, or maybe correlate every pixel with all the pixels, then take the average, things of that sort. So at every location then is going to tell you how correlated is this pixel with its neighbors. And you can imagine if the pixel is inside a cell, it'll be highly correlated with its neighbors, and if it's outside it won't be so much. And so that's a correlation map, and that too has been used in a number of algorithms as a kind of a starting point if you want a way to initialize ROIs or cells for an automated algorithm to take that initial guess of where a cell might be and try to draw something kind of more, with nice boundaries around it. And so that's where, okay, so we go from these like relatively simple ways of detecting cells to maybe like here all the way to the other extreme, where I've kind of labeled this as building a full generative model of your data. And what I mean by that is, one way that you might want to go and find cells in your recordings is you could first write down an equation literally for how you think the fluorescence in pixel I at time t has been created. And so if you have a pretty good model of this, then you can kind of go backwards in this model. So you've written down the math, you have a good model for how, there's some unknowns here. The unknowns are the masks and the activity traces. You can write down your big complicated model, and it produces at the end a movie. And now you have a movie because you've recorded it. So now you can kind of go backwards in this model and try to figure out what the activity was and what the masks were. And so that's what I call a full generative model. And it's along the ways that we started with the initial version of suite2p 2. It's how this, the pipeline that Eftichios and William Paninsky has built. That's called, this stands for constraint on negative matrix factorization. that's incorporated into the Kaiman pipeline that Andrea and Eftikios have built after that. And so this is, there's a few other, I guess, pipelines or approaches of this sort. Now, I'm going to describe suite2p as basically a partially generative model, where instead of, what makes it difficult with the full generative model is if you don't have the right model, you're going to start making mistakes. You're going to start inferring the wrong things. And you will say, OK, well, I'm just going to keep adding more constraints to my model until I make it more and more realistic. Well, now it becomes this kind of big black box of constraints that it becomes harder to know, how does the model arrive at the conclusion that this is the mask and this is the activity. And so really what we kind of wanted to do is to kind of strip down. This approach is good, but we want to control it more. So we want to strip down some of this complexity and just maybe leave in only those kind of parts of the model that really matter for the way we as humans pick up cells, right? Which is we're going to strip down a lot of these constraints. We're going to keep only the important constraints. So things like the fact that cells tend to be localized in space, and they tend to be active very sparsely, and maybe a few other kind of implied constraints, but that's roughly it. So I'll go into more detail about how that works, but that's basically the big picture of these methods. Now, I want to also, because there's not a lot of trust in these algorithms, I would say. A lot of people still like to, they don't like what they get out of the algorithms. They don't understand it, I think, and that's perfectly reasonable because we're all scientists and we really want to understand, where our data is coming from. And when we don't understand it, it's very hard to trust the algorithm. And so the first thing that I kind of want to go through, right. Yeah, well, things are becoming complicated, right? Like it's not, I would say maybe like with calcium imaging, with the kind of stuff we do here, things aren't so complicated, but the more kind of elements you bring to these kinds of problems, the easier it is to kind of lose track of it. And at some point it's going to be, you're going to have to start relying on things you don't understand. It's going to be almost impossible because data sets are exploding, right? The kinds of data that we start collecting and so on. So you just, you want to be smart about it. You want to have controls in place. You want to understand as much as you do, and what you don't understand, you want to be aware of the fact that you don't understand it, right? Because you might get problems from there. Anyway, that's kind of more of a philosophical point. Maybe we can take that up later. For now, I just want to convince you that maybe you want to go by extracting cells by their activity rather than by their structure. So here's from this review paper we wrote last year. Here's a situation where we took the same movie and we took the mean image and we kind of dropped dots at the centers of where we thought the cells were based on this mean image. And you can get pretty confident about this because you see cells and they have this nice donut shape that's unlikely to arise by chance, right? It kind of stands out over the background, so you can really tell that there probably is a cell there, and you draw some kind of boundaries around it, and then we take in this case suite2p, a functional algorithm, and you can see that it finds relatively different things. Some things are clear on both. For example, let's take this one and that one, right? Those are the same cell. Those are very clearly the same. These two cells here, these two cells here. These two are these two, but this one isn't there. And then these other two popped up here, but they weren't here. And OK, so let's try to understand the differences between the types of ROIs that we get here and the types of ROIs we get here. Well, the first difference is kind of a little bit of a circular thing, actually, which is that the functionally determined ROIs have more functional activity, which we quantify by skewness. So skewness is this thing that quantifies how often we've got these outliers in the transients of these neurons' activities. And you can see the blue ones detected from suite2p, they have a higher mean, they're more shifted to the right. The red ones detected just kind of by hand, by us, are really not so different from just taking random ROIs literally at random places over the field of view. And so you might say, okay, but this is circular because you went in looking for things that had functional activity. So of course you found them. But then, really, isn't that what we're trying to find? Are we trying to find signals that are functionally related to action potentials to be able to analyze those in relation to something else, right? And the bigger problem, I think, with this approach, it's kind of described here, where we plot both the neuropil traces and the cell's activity trace at the same time for these different types of ROIs. So neuropil, or background, is just simply estimated from a donut around the cell that we identified. And it's meant to, well, I mean, we all know that there's some of this background contamination in 2P imaging when we record cells or even smaller things. And it has to do with the fact that PSF is kind of elongated, and we're in scattering tissue, so things aren't as, the PSF isn't as nice and tight as it would be with beads. So we always kind of record a little bit of the stuff above and below the cell. That's the background. And we have the advantage that the background is fairly homogeneous across the field of view. So even though we can't see what's above and below the cell, we can see what's left and right, and those are actually pretty similar. And they're pretty similar because they mostly represent axons, like bundles, and not bundles, but like really high densities of neuropil, of millions literally of axons that are going around in all sorts of places. And because they're averaging so many elements, the background tends to represent kind of the average activity of this entire area. So it is maybe the same over a portion of 50, 100 microns, and then it kind of slowly gets less correlated as you go further away. But roughly within the area where we record from, it should be pretty constant around our cell. And so if you look at some of these ROIs down here that were present in our manual label, because we saw a donut there, so we thought, okay, this has got to be a cell. But then there wasn't really anything very clear in the exact same area as a cell in the correlation map, so it didn't get picked up. And we plot the traces from that cell and from around it. You can see that the gray trace, the neuropil, is very closely following the colored trace, which is from the cell that we record. These two cases actually looks like almost the same. This yellow one, maybe there's like, here, maybe there's like a yellow thing that's an event that's actually coming from that cell. So maybe, here, I guess there was something there, pretty dim, but there was a little bit of something on the activity map, the functional estimate. So yeah, there was a little bit of something, but still pretty dim. And so the danger comes that if you're selecting your ROIs from one of these structural maps, you get a lot of things that just kind of represent neuropil, and then you're just studying neuropil for your, as the main component of your study, which is probably not what you want. Okay, and so, right, so that's why we and others, we've kind of all moved to these kinds of functional algorithms to detect ROIs based on function. And I've shown you the neuropil before and how it's kind of decays. The correlation of, any kind of blob of neuropil in somewhere decays like this as a function of distance. So it's super high, like 0.85 or something, right nearby. But then as you go further and further away, it eventually goes to 0.5, and eventually, I guess at a few thousand microns, it's going to go to 0. Okay. And then we can quantify how well isolated these ROIs are from the neuropil. So how much does every ROI that we extract, contain its own signal, as opposed to the signal of the neuropil around it? And we can build kind of these kind of clouds of points where the colored points are from inside the cell. There are pixels inside the cell, and the gray ones are pixels just outside. And so whenever you see kind of a nice clustered separation between them, it means they're well isolated, and there's a number to represent this in kind of multidimensional PC space. But whenever, if you cannot find a dimension across which to separate pixels from inside to the outside, it means there's really kind of no difference between them. It's like as you move from outside to inside the cell, the statistics of those pixels don't change, which is, kind of tells you that the cell didn't have activity. It's really, everything is just this background that we're looking at. And we can quantify that isolation distance here as well. And it's a lot better for the functionally detected ROIs.
Can you say a little bit how you generate the numerical ROI? So you said it's a donut around the cell, but what's the size of that?
Yeah, so the default in suite2p now is to take a donut around the cell a certain number of pixels away, just make sure we're not directly contaminated with the cell's signals. And then you exclude all of the other ROIs detected by suite2p, whether they're somatic ROIs or just like dendrites like you see here. Those are all kind of ignored because we try to, ideally we want to get pixels that don't have a compartment in them, that just kind of represent this big average of things. And so to the extent that we can really get those pixels, then it kind of doesn't matter which pixels around the cell we take.
Right.
So
you exclude other ROIs around
yeah. Sometimes we have to go a little bit further. If the recording is very dense, it might be a lot of these ROIs, so we might have to go a little bit further. Like in CA1. So let's discuss those kinds of examples when we get to the practical. I think that's a little bit of a special case that we should think about more carefully.
Yeah, okay, so let's discuss here.
Yeah.
So one nice way of saying that would be background versus actual cells that are active?
It's their axons. It's most likely their axons, and I'm just, at least that's I don't think there's a measurement to tell us that, but that's what people believe, at least in the literature, that this background corresponds mostly to axons. Not so much to dendrites, because dendrites tend to be kind of thicker. You can even see them kind of by eye in your recordings. Their dendrites, apical dendrites, are these dots coming in through. So we kind of have a sense of what the dendrites look like. But yeah, people think it's the axons and the local axons of neurons in this area. So yeah, it's kind of like the average activity of the neurons in this area. And if you start analyzing that signal, actually, which people doing wide field imaging do, basically, you start realizing that actually it has all sorts of interesting functional properties, like it's responding to stimuli. It has, like in visual cortex, it's retinotopically tuned, actually much nicer than the cell bodies themselves, right? Because it's representing this very average signal. It actually tends to be, a lot less noisy than the single neuron signal. But of course, while it can represent, anything that's shared between neurons locally, it can't represent anything that's kind of more like the salt and pepper tuning of these cells, right? So if you have nearby cells respond to different stimulus properties, well, their average isn't going to respond to that. So that's another reason to want to get rid of the signal as much as you can, is because oftentimes you don't care about the average responses of all the neurons in this area, right? You really care about how some neurons respond differentially from other neurons. The LFP is interesting for its own sake, yeah, for different purposes. Yeah, Chen?
What if, in this case, cells nearby have similar response kinetics?
Yeah, so I think your data is a lot more synchronous than other people's data, so that's why you're asking this question. You will also get something like that in different brain areas. Under anesthesia, you get a lot of synchrony. In slices, you get a lot of synchrony. And so there, in fact, what you'd expect is that these functional methods will start merging together nearby ROIs. I think that's a good discussion to have later of whether you should still be using a functional algorithm for that, or you should be thinking maybe of switching to a structural one.
The other one is how, in which case, what's the ground truth of, what's the criteria of judging if the algorithm already decontaminated the neural signal very well?
Yeah, very good question. If we did, we'd really like to know that, but we're not very sure. The problem that we have, the reason why we can be super sure, is the neural pill represents the average of all these cells, right? But some of the cells kind of represent the average too. For example, a lot of interneurons might, right? They might represent a lot of the average activity of the network. So then the true signal in these cells, in the action potentials, will be correlated to this neural pill signal outside of the cells. Just functionally, not as a contamination, just functionally because they represent, similar variables. And so that's when the problem becomes really hard. Because now you have to rely more on like these cells' transients. So like, all of these, you have to rely on the kind of these outliers to tell roughly how much fluorescence is in the cell versus it's coming from the outside. You can do these scatter plots. I don't think I have them here, but you could scatter plot the gray trace versus the blue trace. And what you'll typically see is you'll see like a cloud of points that represents this baseline. And you'll see like a few points at the outliers kind of sticking out of that cloud. And so there's been various methods developed based roughly on looking at those plots. Like you can imagine kind of doing a robust regression to discard outliers and fit to the cloud. There's no ground truth for this measurement yet, or no very good one. And so it's always a little bit uncertain if what you get out is, if you haven't thrown out an important part of your signal when you do this neural pill subtraction. So unfortunately, I think that's the question that we have fewer answers about. So basically, because of how uncertain we were from that question, we originally had an algorithm like that tried to estimate the coefficients. And in some cases, it would give uninterpretable results, like getting a coefficient of neural pill contamination that's above 1 or negative, right? Like, why would it do that? Well, it would give it above 1 exactly in this situation where the true signal is correlated to the neural pill signal. It should never be above 1 because you should never have more contamination inside the cell than just outside the cell, right? Because outside the cell, well, inside the cell itself displaces some of the volume inside the PSF. So outside the cell, you always get a full PSF of neural pill. Inside the cell, you always get full PSF minus whatever the cell is occupying. So it should always be below 1. So if you're getting coefficients with these automated methods above 1, then it's a problem. We were kind of getting that, and so we decided we're just going to set it to some average value for all the ROIs, and perhaps the kind of things that we do to validate this is we change this value. The default is like 0.7. That's just some number that I think the original GCAM paper used, and then like everyone after that started using. So we just use 0.7. What we'd recommend is you kind of try to change this coefficient, see if it influences your results, and if it does, then you're kind of in trouble, and you have to think a lot more hard about this. But if it doesn't, then it probably doesn't really matter that much. It didn't depend so much on the exact coefficient. Yeah, so that's what I had to say about neural pill subtraction. I think what you'll get a pretty good sense of just looking at a lot of these is, if a lot of your cells look like these bottom ones, then that's not good. That probably means somehow you have a very big neural pill signal. These are going to be your best cells, the ones that have clear big transients. And so whatever effect you're trying to measure, if it's true in these like nice cells that you can give a good quality score to, then it's probably true. But if it's only true in these bottom cells that look a lot like the neural pill, then again, it doesn't mean it's not true, but you have to think a lot harder about where the effect might be coming from. All right, so now to get to the core of the algorithms is a matrix factorization problem. Now, first, we're going to treat, so this is our little volume of a recording. It's got 2 dimensions in X and Y. spatially, and it's got a third dimension that's time, that's number of frames. And so this will be a pretty big object. This will be created by suite2p in this binary file that's sitting on the disk, but it's really just a large three-dimensional array. Here's just some snippets of Python code to just, if you've loaded this movie into, let's say, a Jupyter notebook, you could take its mean, its maximum, you could look at different statistics of it along the last axis of this movie, so the number of frames. I should probably say now, for those of you that haven't seen Python code before, now you don't need it to run suite2p, but hopefully in this workshop, we'll all get a little bit down to the nitty-gritty. So just kind of learning to at least parse this code, right, would be useful, I think, for you. Mostly we'll probably be going through code with you and say, we'll write some little piece of code. It would be good if you could understand what this new piece of code was that we added. So you'll just need a few commands in Python that are a little different from the corresponding commands in MATLAB that you might be used to. But it shouldn't be anything much more complicated than this, really. Okay, that's how the mean image looks like. We've already looked at examples of this. This is how the maximum projection looks like. So I told you that we like this more because if a cell is active even once, it's very big on this projection. So if I go back and forth, you can kind of pick your favorite cell on the maximum projection to see if you can find it on the mean image. And for this kind of high SNR recording, most of them you will find, but there'll be some that you won't be able to find, or at least you won't be certain about them from the mean image alone. Now, this kind of back and forth that I'm doing here is something that we got to doing a lot in the GUI in suite2p. So we have a lot more images like this that you can kind of quickly switch back and forth from, and that kind of really it helps you track the same cell in different kind of views to get more information about is that really an ROI, or is it some piece of another ROI? Is that really two cells? So, again, using our visual system, that's really good to try to identify problems. And then here is this correlation map I also told you about. I'm bringing it up again because we're going to use it to initialize cells. This is just basically a math equation that that formalizes what I just said. We're going to take the average of a pixel with its nearby pixels. That's our correlation map. It's a measure of activity. If you just do that, you can start that a lot of blobs are starting to pop out, right? And mostly in places where there's cells, but also going to be in places where there's dendrites going through and cells that are kind of out of focus and you can't really see them very well on the mean image. They will have a little blob there. Now, this alone isn't enough. Like, you could do one pass to get a number of cells here just by looking at the peaks in this kind of map, right? And there are algorithms that are pretty good at detecting these kinds of peaks. What you're not going to be so good at is detecting a less bright cell, so kind of a dimmer cell next to a bright cell, because the bright cell is going to shine through and kind of increase the correlation of all the pixels around it. And so we'll make it hard to detect some other cell that's right next to it. And so for that, we actually need something more of an iterative process, where we start from here maybe and start picking out ROIs one by one, and we keep peeling off that ROI's activity. So we keep kind of subtracting it off from the raw movie so that it no longer contributes to the correlations between pixels. So that now, the dimmer thing that was another cell, will be the only thing in that area, so we can find it again in this correlation map just with our peak finding algorithm. So that's the basic kind of logic to the algorithm. I told you it requires this iterative, and if you do this iterative process, you tend to get maybe 2 or 3 times more ROIs. That's what's shown here. So this is essentially an algorithm we were using in our old lab in London before we switched to suite2p. It was one that was going through this image just finding peaks. And so you can see it didn't find quite as many as you do if you do an iterative process where you constantly peel off those cells that you already identified to give yourself a chance to find another cell there. Okay, so that's good. We can get more neurons in a more automated way. This was also a little bit manual. Now, to do this peeling off, we need a pretty good model, in fact, of what the cells are, right? We can't just say, my cell is a blob that I'm going to subtract off from there, because if you don't subtract all the pixels of the cells, you'll still get pretty bright pixels left there. And so this is where that kind of generative model comes in, where we want to have a little bit of a sense of how this data was created. And so that sense is just formulated by, again, this equation. This is just a matrix factorization equation, nothing special. We start from this three-dimensional movie, x, y, t. Well, we flatten it because when we're doing matrix factorization, we don't care if a pixel is, exact location x, y. We just want all these pixels nicely arranged in rows. And so now we have a two-dimensional matrix, and we know how to factorize two-dimensional matrices. They're just going to be the product of a matrix that's kind of tall and skinny like this, and another matrix that's wide and less and short. And so one of them is the matrix of masks that corresponds to the masks, the relative brightnesses of pixels inside a cell. If you remember from the beginning, like the nucleus has less brightness than the cytoplasm. So that pattern of intensities in a cell and the exact pixels where it is, that goes into this matrix of masks. And then for each one of these columns, for each one of the cells, or ROIs, there's a corresponding row of its activity in F. And so this is basically the model here is that every single pixel in a cell has the exact same time course of fluorescence. It's just scaled by a different value, by a different weight in different pixels. And so that very naturally leads to kind of these low rank model, these low rank decompositions. It's a, otherwise known as a matrix factorization problem. There are lots and lots of ways to solve matrix factorization problems. The first one for cell detection is actually this Mukamel et al. Paper from 2009 using ICA. This is independent components analysis. This is a matrix factorization that enforces that F, or looks for F time courses that are sparse. And in particular, it tries to maximize their skewness. So we already discussed how the skewness is a measure of how often a cell has transients. Well, that's what this algorithm is looking for. It's trying to do this factorization with skewness constraints on F. And then the constrained non-negative matrix factorization. So that puts some other constraints on our problem. It says, well, we know that both of these should be positive, right? So the fluorescence has to be positive because it's just how much fluorescence is in the cell. It can't go negative. I mean, it can go below baseline of the fluorescence, right? But it's not really negative. You can never have negative photons, right? And then there's a mask. And that mask also can be negative because a cell can't have, pixels of inverted fluorescence. They always have to go up and down together. It can be some pixels go up and some go down. So those are the non-negativity constraints. And then the C part here, the constraint part, that stands for this other constraint on F, which is we don't just want F to be sparse, like in ICA. We actually want F to have, calcium dynamics as close to what we know calcium dynamics to be. So, you go up, and then you kind of decay kind of exponentially, and you go up, and you decay. And so you can put all this information into the algorithm. As I said before, right? You can try to make the simulation as realistic as you can, and then you have a better and better model of this volume. And then you have to go back, and given the data, you have to reconstruct M and F, which, the more constraints you put in, the harder this problem is going to become, the harder to solve, just computationally speaking, and also kind of the harder to interpret if your constraints are a little bit mismatched. So that's this class of approaches. And in the first version of suite2p, we worked on what constraints did we put in. Well, we noticed that we could put just a constrained that the mask should be positive, and we didn't bother constraining F to be positive, particularly because of this neuropil problem, that you have neuropil contamination into a cell, and then it becomes hard to know, what part of the fluorescence is above or below the neuropil. What do you call positive and what do you call negative in that case? And it was also a lot faster to run if we didn't have to impose positivity constraints on F, so we didn't. So we left that out. We put a constraint that the masks should be local, so pixels from the same cell should be localized. And then we also, we thought it would be important to have as good of a model for this background neuropil as possible. So we had a model of neuropil that, actually they already had a model of neuropil that was basically a one-dimensional model that all the pixels are kind of going up and down together. And it was basically, it was kind of like a PCA model of the neuropil. So instead we kind of changed that to constrain nearby pixels to be positively correlated, but to potentially decay at a distance to potentially get different patterns of neuropil, essentially by writing the neuropil in a set of basis functions, so that the bases are kind of tiling your field of view. It turned out in the end that that was almost as good as just kind of high-pass filtering the data. Not super strongly like you saw in Carson's motion registration, just kind of high-pass filtering it a little bit. We know what the spatial distance of the neuropil is, maybe like 100 microns or so. So we should just high-pass filter with 100 microns, and that will mostly get rid of the neuropil. And that's what we do here. Now, let me emphasize in this newest version of suite2p, let me emphasize that this high-pass filtering is just for the purpose of finding the ROIs. Right? We don't want to high-pass filter the movies and then extract fluorescence from them, right? That would be kind of terrible. So we want to be very careful that we keep separate the steps where we find cells and estimate their masks, and the step where we extract fluorescence. Because maybe you don't need to know all the details about how we find these masks. As long as we give them to you and they look like cells, and they talk like cells, you're happy, okay, they're cells, you gave me some cells. But now you really want to know how the fluorescence is extracted from those cells, because that's going to influence your analyses. And so we keep these two parts very separate, the part where we extract fluorescence and the part where we extract ROIs, which is not the case for this approach, for example, at least not by default. But of course, you can always take any one of these other methods and take the masks from them, and then you just do your normal extraction of fluorescence the way you normally do. In fact, that's what I recommend for like any of these. Okay, so there was, so I already told you about one of the updates in this latest version. I should say that roughly the latest version of suite2p corresponds to the Python version, just because we've done most of these improvements after switching to Python. And then this older version, it's also gotten some improvements, 2017, 18. That roughly corresponds to where we left the MATLAB version when we switched over. And so we'll talk a little bit about this new algorithm. Let's talk about it in the context of a kind of a step-by-step through the pipeline, through the cell detection part. So we know we're trying to solve a matrix factorization problem of this sort. M are the masks for each cell, F are the activity traces, and we're doing this iteratively. At every iteration, K, first thing we have to do is we have to find kind of a new candidate cell. And so we basically look for blobs of cells, kind of localized blobs of pixels, sorry, that have large relative transients. And what I mean by that is that they have large, total activity in this set of, 10, 20 pixels nearby on at least one frame, right? So that some frame T here has some kind of average activity in these pixels that's above some pretty large threshold that we set automatically in advance for all the cells. So if we find a part of this image where all the pixels on one frame went high together, very large, that's going to be our candidate for a new cell. And it turns out that, if you do the math and you plug it in there to ask, what is, if I do know the mask already, right? Let's say I assume as my initialization, I assume some kind of blobby mask of just some pixels inside a circle or something. Then what should my F be for that cell? And it's basically just the weighted average of those pixels. And so that's how I estimate the F. And then with this estimated F, I can go in and ask if it crosses a threshold and on how many frames it crosses that threshold. I initialize a new ROI there. Actually, it's with a square rather than a circular blob, just for efficiency reasons, to be a lot faster. Doesn't matter so much. It's just the initialization. And then after I've initialized it, well, now I can iterate within this equation, at this position to get my best estimate of F and M together. But I'm only doing it for this one new cell that I've introduced, which makes the problem much easier than if I was optimizing all the cells together all the time. And so, yeah, optimize M given F, and then optimize F given M. This is roughly how people solve these matrix factorization problems when there's constraints, is they keep fixed one set of parameters, right? Because there's two unknowns here. There's MK and FK, and we don't know either of them. So how are we going to solve this problem? So we keep one fixed, and then to determine the other one is just a linear regression, because we know F. This is our movie. So if we're trying to match the right-hand side to the left-hand side, we keep F fixed, M fixed. F is just linear regression. Now we can then switch over, and we can keep F fixed, and then determine M given F. And it's also linear regression. It's just on the other set of variables. And so if you kind of keep iterating this over and over and over again, you arrive at kind of the best fit for this one new cell that's our one new candidate to be added to our set of active cells. Now, in practice, what I didn't tell you is it's not quite linear regression because we have to keep M positive. So in order to keep it positive, then it's actually just an average of the pixels weighted by the correlation with the fluorescence. It's not a much more complicated step there. It's still part of this iterative procedures, but we have to make sure at every step in the iteration we don't violate this positivity constraint. So that's the kind of only complication. OK, so what we've done is we've had a good guess to start with of where there might be a cell because on a frame it was bright. And so now we've initialized a cell with that guess and optimize it kind of properly to minimize our cost function, our matrix factorization. And then if we do that properly, then we'll be matching as much in the raw data from the cell as we can, right? Every pixel will be hopefully scaled the right amount of the fluorescence of that trace. And so we can just kind of take that term for this new cell and subtract it off from F. And now we have a new version of our raw movie that's now been corrected or peeled off from cell number K. And if we do this iteratively up to cell K, we keep peeling off these different components. And so this is what gives us the confidence. If we're left with something that no longer has activity anywhere, it gives us confidence we extract kind of all of the components in it. Yeah. The threshold changes adaptively based on a few things. One, it changes based on the spatial scale of the problem. So it's actually, what I didn't tell you is that we actually tried different possible sizes of these initializations because different people's data will have different zoom factors, different sizes of ROIs, right? Your ROI might be 6 pixels, or it might be 60 pixels, right? And so the first thing that happens is we identify the kind of the right spatial scale. And from there on, we make sure we try all the different sizes. Now, the critical thing about T is it tells you when to stop. It's not, right? Because we go through these cells, and basically we always take as we go through our iteration k, right, the way we select the new best candidate, sorry, I'm going to go back a little bit now. Okay, so we've got, sorry about that. I'm just completely lost. Okay, so there's lots of good candidates here, right? But the first one we're going to take is going to be the brightest one. And so we don't even need a threshold for that. We're just going to take the brightest one. And so the threshold mostly tells us when to stop, right? We've picked 1,000, and now the next brightest one is below the threshold. It will stop there. And so the way we pick this threshold automatically is by is a function both of the spatial scale of your ROIs, which we estimate, or you can enforce it yourself if you want, but the estimator works fine. And then it's multiplied by the average SNR of these first blobs that we collect from the image. And so basically, as we start peeling off more and more ROIs, it sees that the first 100 ROIs had a brightness that was relatively, I don't know, 100. And so it says, OK, well, I'm going to stop at anything that's like a tenth of this or something like that. We have to look through the code exactly to tell you exactly if it's a tenth or some other way of determining, but that's the rough idea, that it's scaled based on your brightest sources.
Can I ask something?
Yeah.
So where does you're saying that you're estimating that size parameter?
Yes.
So what's the diameter or parameter that we have to tune ourselves?
Yeah, so you could set that, you can impose that. If it didn't give you the right scale.
It's now called spatial scale, not
Oh.
Also, it used to be that in the older version of the algorithm, you need to specify that yourself. It wasn't automatically detected, so setting is still there. It's also currently, so we call this whole new thing, we call, it's still, I think, in a branch.
No, it's in the main version, but you have to call sparse mode 1. So it's not default on, but it's in the GUI as an option to turn it on. It's called sparse mode.
Right. So we found that it worked really well, at least on a lot of our data sets. I think it needs a little bit more, testing, and that's what you guys are here for. A little more testing before we make it the default version. But this is, the new algorithm shouldn't need you to specify much at all, really. Like, you don't tell it how many cells or what spatial scale. It kind of, it finds it itself.
And if you're looking for axons?
It will do something similar, except for this one part that I didn't actually mention it. Okay, so this is our map. It's very similar to the correlation map. It's a little different. Let's say the details are not super important. It's not quite the correlation of a pixels with the nearby pixels, right? Because that doesn't have a concept of sparseness, right? We want to use this intuition we have that if a cell is active on even a few frames very highly, we want to keep it. And so this is instead basically a maximum projection at multi-scale to try to determine the best candidate pixels. So that's how it differs. We still call it a map because we think of it as the map that we go to introduce new candidate regions. But then the other thing that happens after we initialize, so in this kind of inner loop I told you about, where we iterate to best explain the raw movie and to get a good shape for it, there's a process there that essentially grows an ROI from the seed. We identified the ROI at this position, so we know it has to be there, but now we want to get its precise pattern. This is a relatively fancy cell, recorded fairly superficially, so we could get a lot of its dendrites and stuff. Most of them will just have blobs, maybe a little bit of stuff coming out of it. But it kind of, this is how the process goes. It keeps expanding for as long as the pixels that it adds are similar to the pixels that were already there. Basically, that's the idea. Now, I should say, it's not just ad hoc. It's not that we just thought, oh, we should just keep adding pixels that were similar. It's directly minimizing that cost function. But turns out, to minimize the cost function, you more or less are correlating new pixels around with the pixels inside of your cell. So it's also a fairly intuitive process. Yeah, and then if you had something like exons or dendrites, it would just grow from someplace and grow it as much as it can. It has one disadvantage, which is that, which would be hard to see how to fix, but it can, if it has like a weak point where maybe like you have your long axon or dendrite, but it's kind of dim in some place, it won't be able to go past that to get to the other side. So you might get it split into two pieces, and then you'll have to kind of merge those together. And so the reason I'm saying that it's hard to think how you'd fix that is that sometimes you don't want to over-merge things, right? You could also say, oh, I'm just going to merge all the ROIs that have similar activity, even if one of them is here and one is there, right? You still want some kind of spatial criterion, but then deciding exactly if two things should be merged or not, that is perhaps for a later step after you've extracted the ROIs.
You have that parameter called connected or so. Does that relate to this?
Yeah, it does, right. So so there are some parameters for cleaning up these ROIs after you find them. So you find something like this, and maybe for some reason it grew across. I mean, there won't be an example here, but if some of these pixels are not quite connected to each other, they'll be clipped off. And you can turn that on or off depending on. But I don't think it'll do much for this latest version of the algorithm that kind of grows itself organically, because it's kind of naturally restricted from growing over bridges. So that's what it would look like for cerebellum dendrites. So these are all nicely parallel to each other, and so the ROIs are nicely kind of elongated along those directions. Now, there are a few of them here that are split, but it's not so many. It's a minority of them. Basically, that's what they look like. Is this a movie? Okay, this is just another thing to point out here, since we're looking at this data, right? Another way to know your registration worked is if your mean image is kind of very crisp like this. So if you can see like pixel level details, like the synapses on these dendrites, that's good indication that registration worked. Otherwise, things would be more blurry, right? You wouldn't get access to those fine scale things.
And if you have that registration, you'll see it in self-reflection too. I don't know if you said this, you'll see things will be split. You'll see like cells offset.
Right. Because that creates a lot of correlations between pixels, right? So if on some frames, you've got your cell moving, I'm going to stop this. It's distracting. That creates a lot of correlations between pixels from the frames that they're inside. Basically at the edge of whatever ROI you have, there'll be created a lot of variance because the pixel will go up and down depending on whether it's inside the cell or outside. And that can just be all because of motion. And so that creates a lot of variance. And so you'll see it as ROIs that are kind of close to each other and kind of almost like shadowing each other. And you'll tend to get a lot of those, especially if motion registration failed. It won't fail just for one ROI. It'll fail basically for all of them. OK, so here are axons and boutons now, not just dendrites. So these are the ROIs that we get. I mean, in our hands, we don't really get a lot of axons when we label projections like this. We mostly get the boutons. There is the question of which ones of and the boutons, local bouton clusters, which you can maybe see if you look carefully. There are local bouton clusters of 3-4 boutons, but it's not like merging together all the boutons from the same cells. That's kind of a post-processing step. Let's see if we. So this is going to show boutons now. I mean that makes the problem of bouton detection. So because we're not identifying axons so much, it's they're still kind of local small blobs. So it's it's not too different from cells. There's a lot of blobs here, and maybe some axons. Okay. Okay, I think so. These details are just a few things that I left off the kind of main slide so far, but we actually already talked about how we, for cell detection purposes only, to find the masks, the ROIs, we high-pass filtered the movies. One step I didn't mention that I think some of you will want to play with is we actually, every time we introduce a new ROI, we test if it should be split into two rather than just one. And so there is actually a hard-coded parameter there for just how conservative we want to be, and more splitting or less splitting. So you guys could try to play with that, or play with ways to automatically determine that parameter maybe. That would be ideal. Okay, splits don't happen so much. When they happen, it's almost always because it's almost always because there's some kind of bridge in between or some intervening area where the cell or the processes are really not going through, and so it's a spatial constraint problem. So we'll see if there's anything we can do in the algorithm to avoid splits. But if not, I think that's a good scenario for basically an add-on. After you've extracted all these ROIs, you could have a much simpler algorithm now to just merge together things that are super correlated. And again, sometimes in some fields of view, things will just be super correlated because that's what the activity is like. But depending, maybe there's maybe we'll put a parameter in there that will tell us again how conservative we should be with the splits, with merging things back together. So that could be one good thing, and maybe that could be something we do in the GUI, so you can, because I think it'll be a pretty fast program, because it doesn't depend on the raw data, just on the extracted ROIs. So there could just be like a button where you test some level of merging things back, and if you're happy with some level of it, after you've tried a few, then maybe use that one. So I think that's a good thing to try to do in the GUI. So let's maybe try that.
Should say split for how are you going to deal with the overmerges though?
Well, first we're going to try in the main code to see if we can perhaps do better tests if something is overmerged the very first time we collect that ROI.
So you don't want to do it in the GUI at all? Sorry.
I think we could consider that. If that doesn't work, then maybe we come in the GUI and we say, in the GUI the same way there can be like a little program that runs and gives you a new, more oversplit view of the same data set. I think it's like basically there's a slider, right? And there's a trade-off of whether you want things to be more oversplit or more overmerged. And I think if we could automatically figure out that that slider for every data set, that would be ideal. So hopefully we'll get something of that sort. But if not, it would still be useful if you had some slider that you can figure out yourself on at least maybe on one of your data sets, and then it's the same for all of your data sets, something of that sort. The last thing I had in mind were classifiers, which we haven't talked about, and I think we'll just do those more in practice. A lot of times you get all these components and you just want to look at Somata, at cells, and so we make classifiers based on the components that we found, and the classifier gives us the probability that something is a cell. There is a default classifier that we trained on some of the data we had, but you can also build your own classifier based on your own manual curation of a few of these data sets that you have, and that will be, it'll be optimized on your own data as opposed to on someone else's data. Okay, right, so these are some of the features that we use already, and actually we've been thinking for a while of adding some kind of more descriptive features about the shapes of the masks. So I think we're going to try that in this workshop. when it works well, you know, you can, and you estimate that it worked well because, you know, you tried it on some new test data that you haven't trained it on. And then you can get something like, you know, true positives at 95% with very few misses here. Again, this is all based on a human curator, just human curated data sets of how well the classifier works. Yeah, you know, when we get something that is as good as this, we usually for our problems, but this can vary, we no longer do manual curation for the rest of the 5%. We just say, you know, we're not, and it depends a lot on the kind of problem you're working on, right? So sometimes you'll want to be absolutely sure that every single ROIs is a soma, right? Maybe you're comparing different classes of cells and you don't want to be contaminated with the dendrites of another cell or something, I don't know, something like that. So, you know, depending on exactly how strict you have to be in your criteria for accepting ROIs, you know, you can go from taking all of the ROIs and, you know, you've already looked at a few data sets, you're happy with its performance, so from now on you're happy to trust it, or you have to do maybe a lot of manual curation. Maybe you don't have that many cells, but they're really important cells because, you know, it's a very precise cell type and they're very sparse and it's during your, you know, behavioral session that worked and you really have to get every single cell. So, you know, there's a spectrum of between these two scenarios that we try to capture. We try to give you enough options to deal with both extremes. And I think you guys represent a good part of that spectrum. So we'll hopefully be able to refine how well we cover the different situations.
So just to clarify, so let's say if there's a cell that has no transient at all, right? It will be ignored. Is that true?
Yeah, if that cell has no correlation in its pixels, it will be ignored. Yes.
What if that cell, though, say you had also like red channel going on, though, you still want it to somehow be considered as part of your data set? Is there a way to kind of include it based on red that it's included as a non-active red cell? Yeah,
right. So yeah, so we get this question a lot. So I think the right thing to do, right, is not to ask the same algorithm to give you those ROIs, but to run an anatomical algorithm on this, you know, on the red channel, for example, right? So that's the way, because really all you can do is estimate how many cells were completely inactive. So you've got your algorithm that gives you all the cells, and now you have your algorithm that gives you all the functionally active cells. And so now you can compare those two numbers. There is one situation that is for more specialized discussions for like longitudinal recordings. You might have a cell that's not active on one day or one session and then becomes active later. And so you don't want to miss it on that first session, right? And so, you know, the first approach we're going to try with team longitudinal is just concatenating data sets and seeing how well that works, because that will get rid of the problem. If it's active on at least one of the sessions you concatenated it, then you'll extract its fluorescence throughout. If that doesn't work, and it's, you know, it's early days from what we've tested, so we'll see how things go. If that doesn't work, then we'll probably have to do more work in a kind of a post hoc step to deal with that. Okay, I think it's good if I just take more questions now. I don't think there's anything. I talked about how we want to add some shape predictors to increase the quality of our classifier. I mean, you guys don't need to know a lot about this, just that we are working on, you know, getting more benchmarks of these data sets. Actually, that's what a lot of people are doing in the literature. So this is the Kaiman paper. So they had their own benchmarks. There were kind of people manually labeling cells. And you know, there was the NeuroFinder effort before that, started by Jeremy Freeman here at Janelia, to try to you know, create some kind of ground truth data sets for these kinds of problems. The idea is always, you know, the more ground truth data you have, the more you can kind of lead progress, because, you know, coders are good at optimizing things. So if they have some well-defined goal of a benchmark measure, they will probably be able to improve their pipelines. And so that has happened to some extent with these data sets. But then the actual data set you guys are going to be playing with today has in the red channel what we think is a better, you know, ground truth data set. which is it has nuclear D-tomato that is actually put on the same virus, on the same sequence with GCamp. So it's going to go into the same cells. And so right now we can be, basically sure that every cell that has D-tomato, and you can see there's a lot of them, right? These are all nuclei. Remember about how I'm always surprised how big nuclei are. These are all nuclei, and you can see they're very densely covering this field of view. We can compare it with the cells, which are in the maximum projection. So, maximum projection, if you see a cell there, it should be picked up, almost surely will be picked up by suite2p. There might be some cells you can't see here because they're maybe not quite the right shape or dimmer that will also be picked up, but it is a good indication of roughly all the cells there. And so, you go back and forth between those, and what you realize, well, there's a lot of nuclei that you can't see anything over, right? Okay, and so you guys will have this in the example data, and so you'll be able to look at yourself, get more of a sense. A lot of cells are inactive, so that's the thing to keep in mind. They're quite quiet. And that's not because they're not been infected, they're quiet. We talked about neuropil, and so this, I already gave you this little thing that we're really unsure about how to best correct for neuropil because we have an absence of ground truth data sets, so don't have anything to optimize. It's kind of hard. We set it to 0.7 by default. All right, so you can do it on other kinds of data. I think that's all I have for the rest of this, just different kinds of data. And I think this will look like some of your guys' data, like in hippocampus. This is from Waynon, back there. So this is very dense labeled. So this is the kind of data set we'll be dealing with team dense. And here's ROIs. We're going to talk a little bit about demixing. Well, now we're going to talk a little bit. We're going to think a little bit about demixing in the context of these, like when we actually get down to try to analyze this kind of very dense data, we have to really start thinking about overlaps. Like with cortical recordings, we kind of avoided as much as possible to think about overlaps between cells. Actually, what we do by default is we throw out any area of overlaps. We just throw it out. We don't bother to demix it because that's hard and a little bit black boxy. We just throw out those areas. We have plenty of pixels outside of it to estimate the selectivity from. But at some point, your recording is so dense that there's a lot of overlap between a lot of things, and then you can't do that anymore, and you have to think more carefully about demixing these activity sources. So let's see if we get into that scenario with some of these dense labeled data sets. Okay, I also had spike deconvolution, but you know what? This is optional anyway, and I've been going on for quite a bit now. So why don't we take a break? Are we taking a break now?